So to be clear, that for you guys Thursday, for the other for my last two classes Friday, you will have a test. It's a small test, but a test nonetheless. On Chapter 7, cell structure. And function. You're expected to know the organelles and what they do. You're expected to know the cell membrane. And structure and function. And you're expected to know something about osmosis and diffusion. Only a little. Next week, we're going to get into some more complex situations. Honestly, I wish we would have done this 2 weeks ago. I feel like we're about 2 weeks behind what I would like to be. Still not bad. Just, I feel like it was worth the time if it meant that you have some idea, a better idea of what's going on. I don't want to rush things. All right, so you need to know these things. You should have read Chapter 7 a week ago. You'll have this week. Chapter 7 will be due this Thursday/Friday. For you guys, it's Thursday. For my 6th and 7th group period is Friday. So your notes on Chapter 7, you have practice quiz questions you can do. I gave you Quizlet questions. You should know them. They have all the organelles. I gave you those 2 weeks ago. Told you to practice them. Told you to read the chapter, take notes on it. If you haven't, well, you haven't. So as you're going through this next couple days, 2 or 3 days, you obviously need to really pay attention to cell membrane structure because you did this activity last week. I'm about to go over the answers today and tomorrow. And then a little bit about diffusion that will be on the test. So if you know you understand what each organelle does, you understand what it is and what they do, you should understand the types of cells, a little bit about the types of cells as well. So those are the things you should know and you should be able to do. I hope you're able to deal with that. Otherwise, I'm not sure what to say about it, except that, there'll be another test following that. I keep saying this to you guys that it all adds up. You will see it again. You will see this material again and again and again and again and again. We talked about covalent bonds a long time ago. Told you they're going to come up, didn't I? We talked about all the biomolecules and the elements of life. I told you they're going to come up. Well, the elements of life are going to show up again. The biomolecules are going to show up again. And again, and again, and again. So if you learned it the first time, then you're developing. If not, you can catch up and learn it now. All right.
Go ahead. Somebody else.
So
have
I
something
want to be clear
before
when
we
I
answer
say to
this
you,
question.
what is going on? Why is there...
Breathes oxygen. Interesting. So if something breathes, doesn't breathe oxygen, doesn't breathe, is it alive? Bacteria does exchange gases. Some don't use oxygen though. Yeah.
Wouldn't they have to have some form of organs?
Organs. So life has to have organs. Does that mean do bacteria have organs? Yeah.
Yes.
Say what?
Maintains
Maintains homeostasis. That's a good one, isn't it? Yeah. Well, here's the trick. You're all right and you're all wrong. You need a little bit of everything to answer the question. It is mean, but I can't eat the cupcakes, cookies either. I'm diabetic. So if they're going to bring in cupcakes, cookies, and tell me, you got cupcakes and cookies in the dining hall and I can't eat them, I'm going to tell you too. We suffer together. All right, so let's move on. So life. We know life started. Yeah. Oh, it's not on the screen. Yeah, sorry. All right. So life started, as we said earlier, life started around 4 billion years ago. There's a much more accurate timeline present, but I don't have it. My thing ran out of juice and I didn't plug it in. Give me a second. All right, you know what? We'll just grade notebooks, start grading them tomorrow. We'll talk later today. We'll grade notebooks tomorrow. I'll start tomorrow. So it started about 4 billion years ago. And the question is, how did we get from this soup is hot, bubbling, there's a lot of energy, high energy, solution, soup. How do we go from that to the first life? And what did that first life look like? Yeah.
Well, how we got from that to the first life? Wouldn't it be growth and evolution?
Well, how do you grow? That's true. Both are true. Everything you're saying is not wrong. But the question is, what do you need to do if you're going to have life? What do you need to do in order to have life come from this non-living? What's the difference between living and non-living? And one of the things that someone said that was very telling, it's kind of a broad concept, but you have to have something called homeostasis. You have to maintain order, maintaining status quo. There is a reality to the universe that's not very pleasant to think about, but the universe as a whole is moving toward higher entropy. You know this as chaos. The universe is moving towards chaos. That is an observation that is undeniable. Everything in the universe moves towards chaos. Every reaction moves towards increased entropy. Yeah, that means the randomness.
Yeah, do you mind because I've always had this question about entropy. How does one measure it?
That's a good question, but I'll have to get back to you on that. But we can talk during, it's more complicated than I'd like to get to right now, but it's a good question. Yeah.
If we started from chaos and we're moving towards chaos, was there ever a time where everything was just like calm?
Yeah. Now.
Now?
You're it. Your life is a result of that. Life takes chaos and trades. It trades chaos and energy, and it creates organization. So life is going against what the rest of the universe, how the rest of the universe is moving. So this soup is going high energy, going towards chaos. What's the first step to be able to maintain homeostasis, to find peace, as you say? to find order, really, is what we're talking about, order. Your body's constantly moving. It's working very hard to maintain this because its natural state, the natural state of the universe is complete chaos. What is it that in some old texts, in some texts of some Bibles says, they say ashes to ashes and dust to dust? Or dust to dust and ashes to ashes, I don't remember which one.
Like, we all came from dust and we shall all return.
Yeah, we came from dust and to dust we shall return, or something like that. That's actually very true. All things came from dust. Life, I say we, I mean life. The plants outside, the bacteria, all life does is take the dust, the chaos, and create order. That's it. So how do you do that? How do you create homeostasis? How do you create order from this soup? So you got this soup of a bunch of different little things. And how do you take this and put order to it? What do you need to do?
This is going to be very broad. Organization.
Yeah, organization, but you can't. All right, so let me ask you a different way. You have a room. Have you ever been to your sister's room? Sorry, ladies, but this is my experience with my sister. Have you ever been to your sister's room? Those of you that have boys, or that have girls, that have sisters. Even if you yourself are not this way, ladies. If you are, I say sisters, boys are the same. It's just I think what happens with boys. I know in my experience, there were 4 of us, 4 boys and 1 girl in my family, and all the boys I don't know if we just didn't care enough to change our clothes every day multiple times, or we just didn't. I don't remember if we even bathed every day. I can't remember. But, I mean, boys can be a little grody. That's the truth. But my sister's room, I'm telling you, was complete and utter chaos. All the clothes were on the floor. It was literally knee deep in clothes. She used to put a sign on the door that said, "Do not enter. This means you," and she and us. Of course, what did we do? We went in because of the sign. So don't ever tell me no, because I'll just say, "Oh, okay." I'll be right there. So, anyways, my mother would go insane and tell her to clean the room. What did she have to do to organize the room? Organize it. All right, pick it up. She did. She spent hours. What do you have to do? She put it into closets. What she did, she shoved it all into a closet. First of all, I just want to say one thing. That's a really good example. And it really is kind of crazy that you said that, because that's exactly what she did. She literally put all the clothes in, just threw them all in the closet so she can go out with her friends. And my mom opened the closet and all the clothes fell on her. On her. She was not a happy camper. So, but interestingly, putting all the clothes in a closet, lift your head up and participate in the discussion. Hello, if you're sick, go to the office, sign in, please. Somebody tap her on her shoulder and let her know she needs to lift her head up. Take notes. Up, head up. Take notes. What do you say? Bathroom, go. Sign out. So, putting all those clothes in the closet was creating order. It was going to allow her to maintain homeostasis. She had to put everything in the closet. Of course, that wasn't my mother's idea of cleaning or organizing. What's a better way of organizing? Who has a better way of organizing? Yeah. A specific set of okay, and where would you and put them in the closet again?
Maybe
Wash them, fold them, then what? What?
Put them in a drawer, hang them up.
So put them in different. In where? Put them away where? Where's that?
In the drawer?
In different drawers. That's what I was getting to. And look, think about this, guys. I'm just not telling you a story. I'm trying to get you to imagine what's going on here. This is not me wasting time. It's me trying to get to the essential pieces and parts of what life is. Hear me. You're taking those clothes, washing them, drying them, doing all kinds of actions with them, folding them, and putting them in drawers rather than all in one closet.
That's where they came from.
Well, They came from the store, but that's another story. So you put them in drawers. What is the difference between shoving them all into one closet versus putting them all away in different drawers? Compartmentalization. Correct?
Yeah.
That's it. The word is compart. Compartmentalization. Notice I spelled that correctly. And look how long. Now watch. I'm going to misspell the. I'm going to put TEH. I don't know why. I don't know if I'm slightly dyslexic. I have no idea, but I do it all the time. It really annoys me that I do it. I do it, and then I'm like, why did I do it again? The reason that's easy for me to spell is it's spelled the way it sounds. In Spanish, we spell the way it sounds. In English, you don't, because it comes from so many different languages. So it's actually very difficult to remember how to spell words in English. So anyways, compartmentalization is the key. Taking and creating, what is a compartment? It is storage, but when we talk about compartments, that's what this is. It's the compartmentalization is the creation of compartments. All right. So the question, I guess, goes back again. We have this soup. How do we create order from disorder? So you got this giant soup full of a bunch of different things, and you want order, and there's just this chaos of moving particles. How do we create order? We have to create what? What do we call a compartment? Yes, in the case of our example, they were drawers. But in the case of life, what is the compartment called? The cell. The cell is the compartment. It allows you to organize material. Allows you to stop the chaos. To fight against the ultimate disorder. You should be writing this down and drawing it, not staring off into space or rubbing your eyes. I'm just saying, it's going to be tough for you to do well. The cell. The cell is key. So compartmentalization is the creating of spaces. The creation. Why? This thing, I don't understand. Compartmentalization is the creation. Wow. The pencil just stops working. I don't know why. I don't see how. It says it's connected. Oh, it's 0%. That's why. Yeah, it's called my finger. All right. So the creation of space. Spaces, right? Drawers are spaces where you can put your socks in one, your underwear in the other, and your t-shirt and your shirts in the third, etc. Right? So a closet, which was my sister's first solution to her space, to her organization problem, is a space. It did work. It just wasn't as well organized. There was for her. It was just trying to get out of the house so she can go with her friends, but it didn't work. It doesn't work that way. It doesn't work as well as putting it in drawers. Does everybody agree? It's more organized to put things in drawers. So it turns out that that's how life is as well. The first life was likely a single cell. It's something called an archaea. Bacteria. It is something also known as a prokaryote. No, some people spell it with a C, but okay. Prokaryote. There's only one membrane in a prokaryote. One membrane. So that's it. And then inside does have DNA. This is a mistake that a lot of kids make, that there's no DNA in bacteria. They do have DNA. It's circular. Circular DNA. We call these plasmids. The circular chromosome. And then they also have tiny little circular pieces of DNA. I call them pieces, whatever. These are called plasmids. Sorry, I can't write neatly with my finger. Let me try again. So one circular DNA, and they have one big circular chromosome, and then a bunch of little plasmids. Yeah.
What exactly are plasmids?
Just small circular DNA instead of the big one. There's a big circular DNA, that's the bacteria's chromosome, and then you have tiny little ones that are just circular DNA. And then they have all the other stuff. So they just created the first cell was just this soup within a soup, right? This little room, and inside it, we call it the room. By the way, the room cell means a room. When you go to prison, you go into a what? A cell. That's a room. All right. So a cell is a room. And what you do is you create that created the course that someone said it earlier, they have to multiply. Why are you listening? Why do you have those in your ears? Why do you have those in your ears then? I don't understand. I don't get it, but whatever. So prokaryotes are a single room, single space, like my sister's closet shoving things in. And that's fine. It worked. In fact, it worked really well. This one cell became prolific on planet Earth early on. And from North to South Pole, this single cell covered the planet. It was all liquid at the time, or a lot of liquid water. It was a soup. The thing is, it wasn't green. They didn't do photosynthesis the way we do it today. Lift your head up. They didn't have the ability to make chlorophyll, which is what makes plants green. Lift your head up. They did a different process. We still have some of these bacteria around today. We know they're purple in color. So the planet was likely, if you look down on it from space, it would look purple. Because all the oceans would have been purple because this bacteria was purple. It did use the sun. It did photosynthesize. It just didn't do it the way our plants and other photosynthetic organisms do it today. They still photosynthesize using these purple-colored pigments. Yeah.
So was the world like toxic? Was anything?
Well, was anything alive? Let's answer that question. Yeah. What was alive? The bacteria. So it wasn't toxic to the bacteria. Could we have lived back then? No, because there's something else that was missing. Oxygen. There was no free oxygen. There was nothing. There's no O2 gas. All the oxygen was trapped.
So was it like clean? Or
Huh?
was it just like
No, I don't know what you mean by clean. It was great for the photosynthesizing bacteria. For the purple bacteria, it was great. They were happy. But then came along another bacteria, and it was able to take a part of another part of.
So this bacteria just showed up.
Well, in order to understand evolution, you have to understand DNA, and we haven't gotten there yet. So no, they didn't just show up. What they did is they evolved. So the DNA from this bacteria, there was one of its children had a mutation to the chromosome that produced a protein that allowed it to take advantage of a different part of the light spectrum, which made it look green. We call these today cyanobacteria. Yeah,
What does like the G beside O2 stand for again?
gas. Sorry about the messy writing, but I'm going to have to let it go. Cyanobacteria is around today. They are single-celled organisms that do photosynthesis. They are green. They did photosynthesis better. There was a competition. Do you see? Here in the plants that you're growing, you see there's competition. The little ones are competing against the big ones. Only the fittest will survive. So here there was a competition between the purple and the green. The green did it better. So the green took over the planet. There's still some purple bacteria left in different places. I'll show you if you like. Yeah.
What dictates mutation?
It's a really good question. We'll get into it, but to be clear, I'll outline it for you. Number one, mutations happen all the time. It's a natural course of doing DNA replication, copying DNA. Secondly, you can increase mutation by exposure to UV light, certain chemicals. They can increase mutation rates. And certain places in the DNA are more apt to mutate than others. So all those things influence mutations. So cyanobacteria outcompeted, and these guys right here changed the planet Earth. They freed up the oxygen, which allowed all the rest of us to be here. Not just them, but the plants that came after them. And obviously, the least of it is that the Earth went from purple to green. Green and blue now, because the blue is another story, but the atmosphere changed. These little tiny singles, remember what I told you about small things making a difference when there's a lot of them? These microscopic single-celled organisms changed the planet for the better, as far as we're concerned. Yeah. Yes, question, answer?
So in theory, all life on Earth evolved from cyanobacteria and the purple one?
Correct. These were the first. And that is the theory, and I'll show you the evidence tomorrow. Yeah.
So does like science and religion contradict each other?
It depends on what you believe each to mean. There's that word that if you read it, imagine your bedroom without closets, drawers, shelves, boxes, or just a room with a bed. Where would your stuff be? You'd be able to find things needed. How efficiently could you get ready for school in the morning? Would all your school items be set in this study? The compartments you use in your room, the closet, drawers, etc., help you organize items into categories so that you can get dressed in one place. All the items you need for studying are in another place. The compartmentalization improves efficiency. Cells also need organization to improve efficiency. Cells need organization to improve efficiency. Compartmentalization is achieved by dividing up areas in the cell with membranes. Plasma membranes compartmentalize internal structures, while the cell membrane acts as a boundary between the cell and external environment. And 3, if I were going to read that, as I'm assuming you all did, then I would have I made sure I knew 1, 2, 3, 4 facts before I moved on. And by the way, that is life. I would have expected somebody to say cell membrane, cell. All right. So let's take a look at these major components. The first component you see here is this polar head, which includes a phosphate. Now, under normal circumstances, when you see them drawn, what you'll see is a circle with a phosphate in it, and that's kind of the phosphate head. And then you'll see this kind of zigzaggy line. This represents this line here, this zigzag of carbons and hydrogens. That's why they draw it this way. This is known as a what? What is this part known as? A fatty acid is correct. There are two kinds of fatty acids, as you know, and before we go on, let's talk, because you guys had an FRQ that should have been done by now. Hopefully it has been and turned in. A fatty acid has a C double bond O, single bond OH at the beginning. And then there's an alternating carbon, 2 hydrogens, carbon, 2 hydrogens, etc. And we said when we put these together, there's this molecule called glycerol, which is 3 carbons and then hydroxides off of each one. We call this glycerol. And by the way, where is the glycerol in this drawing? It's right here. Oops. This is the glycerol, 3 carbons. And you take a fatty acid head, which is a C, which is a C. Let me do it in white, not blue. C, and I think it's, as I said, double bond O, OH. And then you have the little wiggly alternating carbon-hydrogen bonds. And you're going to get these to bond to something. You're going to get it to bond to phosphate. There's this first one is going to bond to phosphate. So I'm going to flip this just because I don't want to put it on the same side as the fatty acids. But what happens is that you have the OH, and here you have a POH. So phosphate. This connects to some other part you can look at later. That's not really the important issue here. What happens is what comes out when you link these two together. What comes out? Water. So hydroxide and hydrogen leave in the form of water. And together you link them into a phosphate, dehydration synthesis. So you see that phosphate it's also connected to this other molecule here that has nitrogen in it, but whatever. This whole thing here is connected to this carbon through dehydration synthesis. Again, your FRQ requested that you drew that. The second carbon is connected to a fatty acid. Here again, you're going to take this fatty acid. So this, let me erase this other mess that's up here. So one side you have the phosphate connected to it, and again, the phosphate has these other structures connected to it as well. But we're not going to worry about that. We're just going to kind of call it a phosphate head. And then down here, these two fatty acids, one with a single bond, which we call what? What do we call the single bonded ones? That's right, they're saturated. And then one that's in the drawing, one of them one of them is what? Not saturated, but what?
Unsaturated.
Unsaturated is correct. So it has a double bond, which makes it kink out to the side. Again, just a kind of a scribble there on the drawing. What happens when you combine? How do you combine these? How do you make them into a structure?
Dehydration.
Dehydration synthesis. Oops. So this hydrogen and this OH leave in the form of water to make room for the bonds, and then this other OH, this other group also leaves in the form of water, and together they bond into a molecule. This molecule is known, and there it is. This is what it looks like neatly drawn. You can see the O that used to be an OH. This had a OH group on it. Water came out. That's now connected to the double bond O, and there's your saturated chain of carbon and hydrogen. The same thing happened to this middle carbon. And there's your unsaturated. There's your double bond. Your unsaturated double bond. Which kinks, you see how it kicks it out like that? That's going to be important for membrane structure, this kink, this unsaturated fatty acid. All right, let's go to the next. That's all before we even get to the first page of the questions. Refer to Model 1, at least 2 organic functional groups in a phospholipid molecule. Well, we've already listed carboxyl and hydroxide. Anybody have another answer? Glycerol is another one, yeah. Yeah, and amine, that was there as well. Is it time to go? Is that why people are packing up? No, okay. So amine, carboxyl, hydroxide, which one? Phosphate. Yes, phosphate is there, but unfortunately it says this. And when you look at a phosphate, it's just phosphorus with 4 oxygens. We call this inorganic. Yeah. Yeah, it's out behind me. Inorganic. It's the phosphate that makes up your DNA, that makes up your RNA, comes from rocks. They're inorganic. They're minerals in the soil. Very hard. There's a whole phosphate cycle, how that replenishes itself. Phosphate is very difficult to come by. That's why we have to process it, and we use chemical fertilizers to fertilize our fields, in part because of phosphate. So it's inorganic. It's phosphorus with 4 oxygens attached to it. So yeah, but that was good. Phosphate is a functional group. Absolutely important, but it's just not organic. Consider the term phospholipid. What portion of the molecule in Model 1 is responsible for the phospho part? Phosphate, that's easy. What portion of the molecule in Model 1 is responsible for the lipid part? The 2 fatty acids. What part of the phospholipid is polar? Circle the molecule that is polar. It's the head. So if you're going to draw it, it would be. I would circle this head as the polar head. Which portion of phospholipid will mix well with water? Which will mix well with water? Which is going to be soluble in water easily? The head. The hydrophilic, which is not a lipid. Remember, lipids or fats don't mix well with water. So this is hydrophilic. This tail are hydrophobic. These are water loving. They mix well with water. So these are water loving. This is water hating. So one side's water loving, the other side's water hating. The heads love water. Explain your reasoning. Like dissolves like. Let me write smaller. Wow. Okay. This program's starting to freak out again. Okay, it's too small. Wow. Might have to do something about this. One update is all it takes to make everything. So, like dissolves like the.
Okay, just say what you're trying to say.
It's just very frustrating. So you're gonna have to write it down then, without me having to write it down. That's all there is to it. I can't. So the like dissolves like. So the hydrophilic head, being polar, associates with the polar water very well. The hydrophobic head being not the fatty acids being nonpolar do not mix well with the polar water. So because of like dissolves like, in short. Draw a square around the portion of the molecule that's nonpolar. I kind of drew it out there, but I'll try to draw it here. Would this portion of the phospholipid mix well with water? No, we just explained it. Like dissolves like again. The nonpolar material that does not interact well with polar material. The polar water molecules interact with each other very well, pretty much snubbing the hydrophilic part. It's less a repulsion and more of the water molecules attract each other better than they have an interaction with the nonpolar materials. So label the regions of the molecule of phrases hydrophilic and hydrophobic. We did that. I even put a little heart on it. Try to draw a cross and skull bone, but nothing was working. Scientists often use a cartoon representation. Again, this is the cartoon representation. I just kind of did it up there. Like the ones shown below to represent phospholipids. Which portion of the cartoon represents the hydrophilic head? Well, I think that it's in the name, but okay. Wow. Son of a gun. The hydrophilic head, and then there's the hydrophobic tails. So that's what you're dealing with. You're dealing with it when you see these drawings. That's what you should be thinking about. So if you were asked to draw, which is what you're asked, and you have this. Let me draw it again. You have the hydrophobic tail and the hydrophilic water, or the phosphate head. What's going to happen? How are they going to put themselves? You have a hydrophilic head and a hydrophobic tail. How would they align themselves if you put them in water? You know they're not going to mix because the lipid tails won't mix. So how do you get them? Yeah.
Wouldn't it like just be the hydrophilic heads in the water and a tail sticking out?
That's right. That's it. All you have to worry about is that you have these heads in. Oh, I should put them in the water, I suppose. I wonder if I zoom in, will it work? The three hydrophilic heads in the water, and then the two tails sticking out. And that's what would happen if you put, and that's what happens, you get this layer of, when you see oil floating on water, the oil would sit at the top, like the yellow tails are. But the heads stick in the water. And when you have phospholipids, not all lipids have a phosphate head. This is a specific type of lipid. All right. So when the amount of oil is, when a small amount of oil is added to the beaker, the water containing phospholipids will surround the oil droplets, forming micelles. And I would jump right to micelles in the previous video. So use several cartoon representations of phospholipid molecules to show the orientation of the phospholipids in a micelle. So let's think about this. By the way, this is how detergent works. The detergent that you use to get oil stains out of your shirts, you put in your your washing machine. This is how it works. So you got oil, it's in water. You know oil's not going to mix with water, but will oil mix with the lipid parts? Yes, it will. So what will happen is you'll get the phospholipid heads surrounding. They'll interact with each other, no problem. They'll interact with water, no problem. They just don't like oil, so they point away from oil. The tails love oil. Tails have no problem with oil. They are hydrophobic themselves, so they have no problem pointing towards the oil. And they form this little hydrophobic chamber, if you will, hydrophobic cell. It's not a true cell, and you'll see why in a minute. And you should have already, because you should have did yesterday's work. So, and then in the center, you have the oil. This happens all by itself because the water molecules out here love each other. Of course, they're polar, and they love the phosphate heads. But they kind of reject the oil, all right? So you have this thing. It's called a micelle. And it's written right here. Let me be clear that it is here. It says micelle. And it's M-I-C-E-L-L-E-S. I'm just a micelle, not S. So micelle, notice it's spelled differently, and notice it's not as simple as C-E-L-L. Yeah.
What would a broader definition of a micelle be?
A single layer of phospholipids surround, creating a hydrophobic center, with a hydrophobic center. That's it. That's all it is. It's a ball of lipids. Yeah. For what? Over there. Over there. They're over there still. There should be plenty of extra. So it's a micelle. It's not a cell, but this is what happens when if you have a t-shirt or a shirt of some kind. Yeah. My wife would kill me that I can't draw this. I guess that's fine. If you're eating some, I don't know, what do you eat that's greasy? French fries or something, and you get some oil on your shirt, and it's your favorite shirt, you quickly add detergent to it, and detergent is just a phospholipid of some sort. It's a polar head, each detergent has its own formula, and a hydrophobic tail. When you add it
to the stain, and then you throw it in a washing machine, which is full of what? Water. The micelle, the lipid gets surrounded by, the oil gets surrounded by the phospholipids, and then gets washed away, drained away out of the washing machine. That's how detergent works. It creates this micelle. This is three-dimensional. I'm drawing it in 2 dimensions, but you have to imagine that you have like literally a ball of heads in 3 dimensions surrounding the oil. This is a cutaway, so if you imagine you cut this ball in half, and you look inside like cutting an orange or something, what you'll see is inside there is what is it? No, another bee. It's probably literally the same bee. It can't be though. There must be a hole somewhere in the window. It's just a bumblebee. Leave it alone. You're freaking out and you're not focused on the. Yes, go ahead. So 3 dimensions. So you have 3 dimensions. So there's, bless you, there's depth. It's a ball of fat on where the hydrophilic head's on the outside, the hydrophobic center's in the middle, and it gets washed away. Of course, this is at the molecular level. All right. So recalling so what would I expect to see if I were grading this? I'd expect to see a micelle that looks something like this, maybe a 3-dimensional piece, and a labeled, hydrophilic tail. I wouldn't expect a t-shirt example, but that's fine. Recalling the beaker of water is 3-dimensional. What is the 3-dimensional shape of the micelle? Well, I didn't even know that was the question, but there it is. All right, so it's a ball. So it forms a ball. Is that okay with everybody? Everybody understands that? Because there's water surrounding it. The hydrophilic heads are going to go all the way around, and the hydrophobic tails are going to go in the center. So there it is. We're good with this page. Also reasonable. I think you could have answered those. Maybe you didn't. Now you did. Now you're going to upload the final answer.
Wait, where's the package? Oh, you
They're over there. There are. I promise you, maybe they're there, maybe they're over here. Maybe you can go make copies, but I swear to you, there's extra copies in that. Did you check behind the plants? Okay, because I'm telling you there were at least 10 extras. And I don't know, I printed 150 of them. I only got 120 kids. Are you guys eating it or something? Yeah, those are, the front is, not the back, not the rest of it. I'm sorry, I just don't have it right now. If it's not here, it should be extra copies. There were plenty printed. All right, so when we're looking at this, and again, just to be clear, the difference between when we look at this and you see this one, this has a hydrophilic, I see. A hydrophilic outside, the heads are facing out, and then you have the hydrophobic inside here, the face is inside, and in the very center, you're going to have oil. Outside, you have to have water. That's when this happens, as we looked at in the previous slide. That's number 1. Number 2 would happen when you have oil on the outside, but on the inside, you would have water. Because the hydrophilic heads are pointing inward, you'd expect to find water on the inside, and outside you'd have oil. That's in the second situation. In the third situation, what you'd expect to see is water on the inside and water on the outside. And how do I know that? Because you can see the hydrophilic heads.
They're on the surface, right? What's the hydrophilic heads going to be pointing to? The water. They have to be pointing to its water. Because if it was oil, they would be repelled by it. They wouldn't form an organized structure. And so when we look at here at the inside, we'd also see water. Because the hydrophilic heads are pointing in. And in between, you have two layers of fatty acid tails of lipids, right? So we have phosphate heads and lipid tails, two layers of them, where the lipid tails dissolve each other. We call this a phospholipid bilayer. Two layers. Phospholipid bilayer. Everybody okay with that? All right. So the example, and that's what a cell membrane is. A cell, if you've done your work, if you did your work yesterday, if you read your chapter, you know you have a cell that has a cell membrane. And right now, I'm going to look at just a prokaryote. What is a prokaryote? Just a single cell membrane, no internal membranes, so we'll look at that in a minute. But if we took a close look at the cell membrane, what we find in a cell is inside there's water. Outside there's what? Water. I hope everybody realizes that now. If you didn't before, I hope you realize it now. So that means there has to be hydrophilic heads pointing inside and hydrophilic heads pointing outside. And then in the center, there has to be hydrophobic tails. There has to be, because you have water inside and outside. Because this, one of your colleagues just said a little earlier, if like dissolves like, how is it possible that you could form a membrane if the lipid tails are repelled by water? Well, it's the hydrophobic heads that point towards the water that allow this membrane to exist. Yes?
So that white line is the cell membrane?
Is a cell membrane, yeah. It's the cell membrane. I just drew it in yellow just to show you the different colors. But yeah, that would be called the cell membrane. That's one name of it. That's one version of it. Another name for the cell membrane is called the plasma membrane. Another word for it, or term that represents it, is a phospholipid bilayer. Now, I have to say that this is a phospholipid bilayer. This is a cell membrane. In a prokaryote, that's the only membrane it has. Yes, question?
So, the part where the hydrophilic tails are, what's inside there? Like there's no water. Is it just air?
No, it's the tails. The tails are in there. But that's a good question. That brings us to what else is in that membrane. So that's going to be an investigation we're going to look at later. But just to give you a clue, you have the hydrophilic heads on one side, hydrophilic heads on another. And we'll talk more about this tomorrow since we're not having the test. And you see that what you have on the inside of the membrane is what? You have these, the lipids. Now, what else might you have in there? You could have this I don't know how to draw it, but we'll just go ahead and draw it like this. This thing known as cholesterol. You've heard of this?
Yes.
Cholesterol, oh, is it frozen? Oh, this is really, I'm gonna have to buy a new app because this app is just, it's not playing well with the new.
Hey.
Somebody say something to me? Yeah, no, it came off because I turned it off. I had to turn it off and turn it on. So
the phospholipid bilayer, in the phospholipid bilayer, you can have, is it recording? Yeah. In the phospholipid bilayer, you can have cholesterol. In fact, you do. And in animals, cholesterol, by the way, cholesterol is only found in animals. So that's something that's, when people tell you no cholesterol-free sunflower oil, all plant oil has no cholesterol. Cholesterol is not found in plants, only in animals. Cholesterol helps keep the membrane fluid. Does everybody know what fluid means? Can someone tell me what fluid means? Good. To flow. Who said that? To flow. That's right. That's intuitive. Very nice. Well, before we get to that, do you know what fluid means? So fluid means to flow. What kinds of things flow? Who can tell me? Liquids for sure. Blood is an example. Correct. So liquids for sure flow. What else flows? It's not just liquids though. Gases, yeah. Gases flow. If with liquids, you know they flow because you've seen rivers flow, right? Or creeks, or a stream, or water going into the sewer. How do you know gases flow? Wind, yeah, wind. You can feel the flow. When you stand in the wind, it's like standing in a creek. It's the flow of the gas going across your body. It's flowing. It's a fluid. So the membrane needs to be fluid. It needs to be able to change and move. It's constantly moving. And this is a fluid. It has, even though it's not water, it's a fluid. They stick together because of the interactions between the bilayer and the interactions, or rather the hydrophobic interactions here and the hydrophilic interactions here. These stick together. The polar molecules stick together, correct? And the nonpolar molecules stick together. So the membrane holds itself together that way. That's an answer on that handout, by the way. One of the questions on the handout is, how does a cell membrane hold together? How does a micelle hold together? You should be answering that. So you can almost consider these as guided notes for you. So the hydrophobic interactions keep the hydrophobic part of the tails together. The hydrophilic interactions keep the hydrophilic heads together. So that's how membranes stick together. That's how your cells, you're made of cells. Why don't they just fall apart? Because they're being held together by those tiny little interactions between the molecules. Now, what happens when it gets cold out? What do fluids do when they get cold? When it gets cold out, they freeze. They come together too closely, and once they come together too closely, they become a solid. We don't want to become solid, do we? So what animals do is they add this cholesterol molecule to the membrane, and they also add another thing. They do is they'll add an unsaturated fatty acid. So if the more unsaturated fatty acids you have, the more space that there is in between the molecules. Does that make sense? So that pushes the other phospholipids away. So in the winter, what you're going to find is more unsaturated fatty acids and more cholesterol in animal membranes than you would in the summer, so that the membranes are less dense. This is an exaggeration, okay, just to make a point. You get more space in between the molecules, so when they start to slow down, they don't freeze into a solid. They act almost as an antifreeze. Yeah.
Is that why people's skin kind of dry with?
That's a really good question. Why is the skin dry in the winter? No, that is not why, but it's a really good observation. Okay, I just want to point that out. The reason your skin gets dry is because the air is drier and more water leaves your skin, so your skin gets drier. That's why putting waxy coatings like lotion on your skin stops your skin from drying out. Okay, so that is different. Yeah.
If you went to the doctor
in December and then went back in like April or like towards the summer and they took like samples of your blood each time, would they look different since you said something about
You make me almost want to cry how beautiful a question that is. That really is a beautiful question. I want to point that out. I know you all think beautiful is, all these styling things you all do, which you are, very beautiful. But for me, those kinds of questions, the kind of question you two just asked and observations you're making, it just shows how beautiful your minds are. Okay, just amazing. Absolutely right. So when you take blood samples, the conditions under which you take them can influence the results. So humans, we don't live in the cold. We're not, right? In the wintertime, you're not out there, are you? You're in the house. You're walking around. You put coats on, so you don't your body doesn't react as intensely as like a rabbit that's living outside trying to forage for food in the middle of winter. Correct? So they need to keep their blood a lot more. Their cholesterol and their blood samples is probably going to be much higher than yours might change in the winter versus summer. But yes, animals do change their cholesterol levels in their blood. And by the way, you make cholesterol. You don't have to eat it at all. You can not eat cholesterol at all, and you will make it. Okay? To my understanding, as of the last articles I read, and I'm no doctor, so don't take my word for it, how much cholesterol you eat has very little impact. Dietary cholesterol has very little impact on your cholesterol in your blood. The impact comes from saturated fatty acids. As we see, saturated fatty acids can make your lip membranes more stiff. So your membranes become more stiff with less cholesterol, more saturated fatty acids make your membranes stiffer. More unsaturated fatty acids, more cholesterol makes your membranes more fluid, more space, right?
So it's possible to be wrongly diagnosed with high cholesterol?
No, it's a good question. Unlikely. The difference, as I said, with humans, it's a little different. Our bodies don't change that much because we change our behavior to deal with that freezing problem. Other animals that don't have the ability to do that, they might grow thicker fur. Animals shed in the spring, in the summer. They shed because it's hot. Then they grow new fur for the fall and winter. Does that make sense? We don't do any of that. So we have a brain that allows us to change. So can you get a misdiagnosis? Yes. Would it be because of that? No. Yes.
You said higher cholesterol can thicken blood, so is high cholesterol
No. Makes blood less, makes the membrane more fluid, not thicken it. Doesn't thicken it. And thickening the blood, when we're talking about diabetes and increase in blood pressure, that's literally because sugar is polar, sugar is sticky, very sticky. It changes your blood, enough concentration will change your blood into something like syrup, which that's different. That's dissolved polar molecules that thicken your blood, make it more viscous because everything's sticking to itself. And that causes all kinds of problems. So yeah, diabetes is mainly a problem of sugar, which is a polar molecule. Okay, saturated fatty acids will make the membrane more stiff, unsaturated more fluid. Are we all good? Not that I understand. No. You should talk to your physicians, though. Don't be changing your diet based on what I say. I'm just a high school biology teacher, but my understanding is no. The problem comes, though, is that with things that are high in cholesterol are also tend to be high in saturated fats, and they tend to be high in trans fats. So like bacon, high in cholesterol, also high in fats. One good thing that's high in cholesterol but not high in saturated fats, eggs. You used to think eggs were bad for you. Today we're pretty sure, from what I've read, that eggs are good for you. So whether that changes when we learn more, I don't know, but that's my understanding thus far. All right, so are we all good with this? All right, so now that's prokaryotes. Interestingly, there's another group. Let's talk about some of the stuff you had yesterday. There's two groups. There's three big domains. We call them domains of life.
three big ones. There's archaea. That's that's a type of bacteria. And there's bacteria, I'm just going to call it bacteria. And then there's eukaryotes. I think it's Ada. Is it Ada? I think it's Ada. Eukaryota. Eukaryota, bacteriota, and archaea. These are three domains in life. These two here are what we call prokaryotes. Which are the ones that we just described above, okay? And we'll describe them better later, but bottom line, just one membrane. That's it. They might be that shape, they might be round, they might be that shape with something called a flagella, which is an extension of that membrane, but it's just one membrane, regardless. Inside these, you will have a circular. It's a double helix. Remember what the double helix in it's on your school crest, right? The double helix looks like this. In eukaryotes, there's an end. There's a 5' end and a 3' end. In bacteria, there's no end. It's a circle, but there's still double helix.
It froze.
Did it freeze again? Oh, okay. So I wouldn't be surprised. I'm going to draw two colors. So you see it's a double helix, but it's a circle. Do you see that?
Yeah.
All right. So it's the same with all the various types of bacteria. They have a single chromosome that is circular in a double helix format, and that's how bacteria do their DNA. They also have smaller double helixes that are smaller pieces of DNA that also wrap around each other in a double helix. And these things are called plasmids. We talked about that yesterday. Are we all good with that? It's time to go. Well, before we go, do you have time to write out the rest of the organizations, just 4 or 5 titles? Is that okay? Yeah. All right, just really quickly. Eukaryota, you have, in eukaryote, you have fungi. And we'll finish this tomorrow. Fungi, you have animals. Yeah, and the next one's plants, is very good. Does anybody know what the last one is? There's 6. So far, I only got 5. The last one's what? You guys know from SpongeBob. Not Patrick. Patrick's an animal.
A snail?
Snail's an animal. What? Squid's an animal. Yeah. Sponges are animals. Good guess, though. Huh? Single cell, one eye. Plankton, yeah. They are protists. Plankton are protists. So these are the 6 kingdoms of life. These are the 6 kingdoms. Archaea. Make sure you know them for the next test. Animals, fungi, bacteria, archaebacteria, plants, and protists. All right, very good, guys. Good job today. So under fungi, there are these groups of life that you know of, that you've seen in the.
That's a mushroom.
A mushroom, correct. Badly drawn mushroom, but still.
Oh, you recorded this? He said that was
So mushrooms are fungi. So are these little things called yeast. We use yeast to bake bread. We use yeast to make beer. We use yeast in a lot of things, and yeast can cause infections, yeast infections and urinary tract infections, and in males and females. Huh? Yeast, same yeast. They're different strains. So yeast, yeah, they're
Oh
yeast.
yeah, Mr. Mendoza.
Baker's yeast is different from the yeast that cause infections, but they're still fungi. They're still same group. Yeast are yeast. There is mold. Mold is also. I hope you're listening, and I know you're not.
I am.
Not you. So you have.
That's not you.
Could you all back there? Shh. You have yeast, and you have mold. Mold in the bathroom. This mostly single cellular is also fungi. Black mold is just mold. They're just all fungus. Athlete's foot.
It's a whole different.
They're different kinds of. I mean, obviously, they're different kinds of yeast.
Yeah.
Sorry, different kinds of fungi. It's not. They're not all the same. And you know something called ringworm. Ringworm is not a worm. It's a fungal infection. Dandruff. I can't spell dandruff. Somebody spelled. It's the dandruff is the result of a fungus. Dandruff is caused by a fungus. At least some of the forms of dandruff. I can't.
What if your scalp
Whatever.
is
I think it's
dry?
UF.
It's Mr. Mendoza, what if your scalp is dry?
If your scalp is just dry, a little itchy, that's not the same as having dandruff. Dandruff is when you have a lot of dead skin, and it's like that's thick and it falls out. You could make it snow on your desk. Then it happens. It's a light fungal infection, easily cleared up with some dandruff shampoo. It's not a big deal, but it is in fact a fungus. Now you have, or at least it's caused by fungus. Excuse me. Animals. I can't even go into it, but there's birds. There's birds, which are dinosaurs. I hope you know that by now. There are mammals. There's herbivores and carnivores and mammals as well. There are fish. There's sponges. And there's a lot, right? There's a lot of different animals. I'm not going to go into all of them. Each one has a different sort. Obviously, something like a squid and an octopus is different from a fish. So that each has their own way of doing things, but they're all animals. They have common characteristics that make them animals. We have plants, yes, sponges, like SpongeBob. It's an actual animal. Did you not know that?
No.
Yeah, but it is. They actually do exist.
Oh my God, what place?
In the ocean, yeah. Say what? Yes. These will be on the test. Plants obviously are things like grass and trees. There's a lot of conversations going on, but whatever, flowers, cacti, seaweed. Seaweed can be algae. Depends on where you, but seaweed, let's go with seaweed. Let's call it kelp. I think kelp is a plant.
Yeah, kelp is a plant.
Huh? Yeah.
Did you try seaweed before?
Yes, I love seaweed. I love seaweed. Yeah, I eat it. They're all salty. They're from the ocean. So proteins are things like algae.
Excuse me, Mr. Mendoza. She
Can-
got a question.
Yeah, in a second. Algae. I just want to finish this before class is over. Algae. No, barnacles are animals. Algae. Plankton. Plankton is real. Proteins are real. There are plankton that are animal-like. There are plankton that are plant-like. Some do photosynthesis. Some are multicellular. There are things like amoebas.
What's a meat?
There are ciliates. So amoebas look like something like this. And they have amoebas go and surround and eat things. Ciliates have little cilia on them.
They have small mouths, they have multiple nuclei, that kind of thing.
What's the third word?
Silly.
Ciliates, amoebas, plankton. Plankton they don't have a single eye, but they actually do look a little like plankton, and they kind of look something like this. This is not an eye, that is a nucleus. If plankton were if the cartoon were drawn from a real plankton, it would be a nucleus. So some plankton have tails, they swim, others don't, so it depends. Yeah, they're plankton, all the things in SpongeBob are real, but obviously they're made up and exaggerated. The characters are made
I
up.
thought they was talking about a sponge, like from that kitchen.
No, the sponge from your kitchen used to be sponges from the ocean. That yes,
Everybody get them.
they were animals. Sponges used to. Excuse me. Let me finish before you freak out. Sponges from the ocean used to be used as sponges in the kitchen. We no longer do that because we can make them synthetically. We don't need to collect them from the ocean the way we used to. They used to collect them, dry them out, and then they would be used, their bodies would be used as sponges. And you can go get one. You can get them. You can actually buy them on Amazon.
Okay.
Yeah. Look, you can buy almost anything on Amazon. Yeah. Yeah, Venus flytrap is a plant. It does photosynthesis. It only captures flies and other insects in order to get nitrogen because they live in nitrogen depleted areas where they don't have a lot of nitrogen. Yes. Huh? I'm surprised that you have this idea. Let me be very clear. We used to use whale oil for our lanterns to light our streets. So that, what is whale oil? We would collect the whale and they'd cut off the blubber, and they'd melt it into a liquid form, and they would put that liquid oil into lamps and we'd burn them. That's how we kept ourselves warm. To write, our tools were made from bones of animals. Or the tusk of an elephant. Or we use feathers as quills. That's another example.
Why did they leave out stuff?
Say what?
Why did they leave out stuff?
Why did what now?
Why did they leave out stuff that y'all use for that people?
Wait, I don't mean leave it. Wait, I'm trying to understand your question. Leave it out what? Why is this now? Lipstick today is not made of anything except wax and coloring. Wax used to be made of beeswax. We would raise bees. We then, at the end of the season, they would take the honeycomb, squeeze out the honey. That wax would get melted, and you could make them into candles. You could take that wax and make them into lipstick, that kind of thing. Today we have synthetic methods of making wax, so we don't have to depend on bees. Maybe it was made out of whale, but I don't know. I don't know specifically. I know that waxy materials usually come from things like beeswax. You're sitting on a dead thing right now.
Wait, beeswax like the
The tree, yeah, the tree was dead. The tree was alive. You're sitting on its skeleton. Why is that funny? It was alive, and you killed it and cut it up. Somebody did. What?
Like that's different.
Why is that different? Because why is a tree different?
They had no.
It can't move, so it's okay. So if somebody's paralyzed, it's all right.
No, I didn't say
No,
that.
I'm just saying a tree can't move. That doesn't make it any less alive. Now, I'll grant you, it probably doesn't feel much, but it does sense pain. Trees do sense pain. They do react. They do communicate. So we know that with chemicals. If a caterpillar is eating the leaves of one tree, they send signals to the neighboring trees, and those other trees react. They're chemicals. They release chemicals into the air and through their roots. They react by producing poisons that kill caterpillars. So if they start, if the tree, in some cases, obviously, other cases they might not produce as much, as many leaves, not produce as much fruit, whatever it is that's going to reduce the infection. Yeah, it's interesting. They don't have brains, true, but they are alive. They're very much alive. They're feeling, they react to the environment. Some people report that plants react to music, soothing music, versus, I don't know, I don't really know if I believe those reports, but that's what they used to say. So, but the point is, for sure, they react to their environment. They are alive. So just because they can't talk, they can't move, doesn't make them any less alive. In fact, the fact that some of these trees are 300 years old, 1,000 years old, and we cut them down for wood, that makes it somehow a little more intense, doesn't it? It's like some of those trees were alive when Jesus was walking the earth. Do you ever think of that? Some of those redwoods were alive when Jesus was walking the earth, and we cut it down for trees, for wood. All right, guys, have a good day. So in number 14A, what would the answer to that question be? What happens if phospholipid tail when you have a double bond? I've said it like 5 or 6 times. Yeah, go. And what?
It becomes more flexible.
The membrane does become more flexible because of the double bond. You're right. But what happens to the tail?
It gets bent.
It gets bent, is correct. So instead of having single bonds that would allow the carbon chain to be straight, again, this is a cartoon of it. When you replace one of these saturated, these are saturated fatty acids or single bonds. When you replace them with a double bond, you have an unsaturated fatty acid which means it's bent. The tail is bent. It points out. It takes up more room. And if the membrane's made of a lot of unsaturated fatty acids, it's more fluid. More fluid. Somebody said it's less stiff. Is that okay with everyone? Everybody understand that? So saturated fatty acids and membranes make them stiffer. Unsaturated makes them more fluid, the membranes. But it's because the accumulated effect of having a lot of unsaturated fatty acids in the membrane pushes the phospholipids apart from each other. Explain why the flexibility, fluidity of membrane increases with more phospholipids in the layers that contain double bonds. I think I just said that, right? Yeah.
So this is off topic, but this is just for me and Zai leaving. We're gonna be leaving at 9:15 for the office. Just let me know that.
9:15? Okay. Yeah. Do you guys have a pass?
Oh yeah, my pass. We do not. No.
Okay. They just told you that?
Yeah, I was miscorrect.
All right, that's fine. I just need to know that they give you a pass or not. So I'm assuming you don't need a pass from me, so go. You can go right ahead as soon as you're ready. So when you have the double bond in the same number of phospholipids, the phospholipids are not as crammed in the same space. Does that make sense? If they're not as cramped, they have more room to be, they're more flexible. The membrane is more flexible.
Are you okay?
Yes, some dust in my throat.
You need some water?
No, thank you. I have coffee. So when we're looking at when you have saturated fatty acids versus unsaturated fatty acid, you have more flexibility in the membrane because there's more space in between the phospholipids. All right, so moving on. And we're good with all that? You wrote it down and you're just not drawing it? Because what I'm noticing in notebooks, when I grade notebooks, is people will draw that. They'll have that. Sometimes they're labeled, sometimes they're not labeled. But then there's no meaning behind what they draw. So you wrote that answer, but what would you write? What would I write if I were taking notes? Cornell, somebody came to me this morning and they said that, you know, they're going to be this, hopefully the student council president, and that they're certainly involved with student council. Some of their constituency came and said that, you know, things could be a little more organized with the Cornell notes on the board, that we're kind of zooming in and out and moving in and answering different questions, and that's all true. But each individual part of the notes are presented in a very organized fashion. At least I try to. And if you have this opinion, I more than respectfully would ask you to stay after school with me, or come in early, and we could sit down and watch the video, and you can show me what you mean. And I'll show you what I think is going on. And then we can decide how we can improve it. Is that clear? Yes? Do you hear what I'm saying? If you think that things can be a little more organized, then let's sit down and brainstorm on it. All right? But we'll have to work on that at after school. I'm here again today we cannot stay after school. They're going to have end of pep rally, everybody goes home. Is that clear? So I'm going home right after pep rally. You should as well. If you don't want to be, a lot of people have anxiety with noise, and often what I'll do is I'll have kids sitting here playing chess, relaxing, doing whatever they want to do quietly. You can be here with me if you'd like during pep rally. That is an option if you're having anxiety. If you've never been to a pep rally, it's very loud. The entire school will be there, all 1,000 students. It's very interesting. It's fun. It is fun. I don't like the noise. Noise bothers my ears. That's just me. But if you want to play chess and have a rematch, so I can get back at you beating me, you're certainly welcome to stay here after school. Yeah. No? All right. Thought that was a question. All right. So if you're writing this and you do what's on the board later tonight, or in your study group, or doing a tutoring session, you might, or even now in class, if you hear me babbling about something you don't really care about, you might want to go ahead and write down over here in the main points that, you know, the summary of this, right? So the main point here would be that the unsaturated double bond increases flexibility of the membrane because the double bond bends the fatty acid so that's what I would write on the side. So I have my note now. Of course, this is a handout, right? So you might actually write that here, right? Huh? Which one? There were a whole 140 copies of them made now, and you didn't get one. I don't know what to say. You can stay, as I said yesterday, for people that said they didn't have one, you can stay after class and I'll give it to you.
Are you doing that for today too?
Yeah, I mean, you guys gotta make the effort. Yeah, I'm here. I'll make copies. All right. Now let's move on. The diagram below shows that the chemical structure of cholesterol, which is a key component of the membrane, which is a key component of membrane structure. So cholesterol's in animal membranes. Animal membranes. So that's something that you would probably want to add. It's in animal membranes. So you're not going to find them in cholesterol in yeast or in plants or in other organisms. That they're not going to have this cholesterol. So is cholesterol molecule mostly polar or mostly nonpolar? Nonpolar. Circle the drawing which would illustrate where you would find this part here might stick in the right here, that might stick out into the towards the out, but the rest of this whole giant molecule, it's big, is nonpolar because they're all carbon and hydrogen. They're all carbon and hydrogen. So that's nonpolar. It's a lipid. It's a lipid. Also, so this is a lipid. All right. So cholesterol, how would it be oriented would it be oriented this way? If this is the membrane, this is outside the membrane, this is inside the cell, this is outside the cell, this is the membrane. You notice, please notice, please notice, let me highlight it, that you have a polar heads on one side, polar heads on the other, polar heads on one side, polar heads on the other, polar heads on this side, polar head on the other. You have in the middle, please, again, try to make sure you understand that you have two layers of lipid tails. So because you have two layers of phospholipids, what do we call this? That's right, we call this a phospholipid bilayer. And you see that this is outside and this is inside. All right, outside and in, phospholipid bilayer. All right, where would you expect cholesterol to be found then? Yeah, anybody? Yeah, go ahead.
Inside the bilayer?
Inside the bilayer. You'd expect this giant nonpolar thing to be in the nonpolar area of the... So this would be wrong because this would be... What's outside? Just to remind you, outside is mainly water, H2O. Inside is mainly water, H2O. And yes, bless you. So on the outside, you would not expect lipids to be dissolved in water, so this would not be a correct answer. This would not be a correct answer because why would it hang out? It would still be in water, right? And this wouldn't be hanging out. So this is the right answer. This is it. All right. Cholesterol forms weak attractive forces of multiple phospholipids in the bilayer. Would this increase or decrease flexibility of the membrane? Explain. All right. So with that, I'm going to let you answer it, and then we'll talk about it again. Oh, you have an answer. Go ahead.
Yeah, this is just a hypothesis. I think it would decrease it.
Decrease it?
Yeah, I think with the increasing bonds comes the decreasing flexibility. I think it would give enough space. That's just the hypothesis.
You say decrease flexibility or decrease?
Yeah, it would decrease. Yeah, it would decrease flexibility.
All right. So we're gonna go. You're gonna do a mini research project as soon as we're done with the notes on the next page, and you guys are gonna try to figure that out, and you're gonna explain it. And it'll be in groups, and I'm gonna want to know an answer. Before we go to the embedded questions, or do you want to go into the extension questions first? What would you like to do? The extension. So let's take a look at cholesterol really quickly. I said, and I told you this, that cholesterol has a polar head. That's what allows it to kind of be at one side or the other of the membrane. So it does have a polar OH group. It's mostly nonpolar. It has a steroid portion. These multi-rings are called steroids. So it's a steroid portion and has a hydrocarbon tail. This is nonpolar, but it does have polar side. We call it amphoteric. So it's both polar and nonpolar, just like phospholipids. So, except this is only slightly polar because this group is smaller than a big phosphate group, right?
Yeah.
All right. So you're kind of right. And the word stabilization is interesting, but not very complete. Yeah.
So, I had that,
Shh.
I had that increased flexibility by stopping the fatty acid tails from sticking together, which would open up the area.
That's right. It does that. So it does, and that's what I think I told you in class. It separates so what happens when it gets colder in the wintertime, especially for animals, is an issue. It gets colder in wintertime, the phospholipid the tails are all very hydrophobic. And they get slower and slower, and as they start to lose kinetic energy, they get closer and closer, and they kind of start sticking to each other, and they get stiff. The cholesterol, being nonpolar, is embedded in the membrane, keeps that distance, keeps that from happening. So that's one way it stabilizes the membrane. Interestingly, that also in that membrane, that cholesterol will also not only keep it fluid and cold, it also does have that it does kind of stick to some of the phospholipids, creating stability even when it's warmer. And it makes it less permeable. So it's almost like putting a waterproof coating on your cell. So it also makes it less permeable to random molecules. So it does those three things to stabilize the membrane. It does attract the phospholipids, so it makes it more stable. It does increase fluidity, and it does make the membrane slightly more waterproof, slightly less permeable. All right. So it does those three things. Cholesterol is a very important animal. I hope you wrote those down, because cholesterol does have a steroid part. So to be clear, this portion here is, I'm talking, I don't know if you hear me, but, hello, I know you're not hearing me. Some people are not listening, so listen, please, with sugar on top. Double please, with double sugar, I don't know. So this is steroid. There's a steroid part, you never heard it. It's, all right, whatever. This part here is a hydrocarbon. So, as I said, this is nonpolar, but this is a hydroxide. So these are more functional groups, right? They tell you how they might function. This is going to be polar, so it's going to kind of stick in the polar part of the membrane. This is nonpolar, stick in the nonpolar part. This is very, very water resistant, very, very nonpolar. So what it does is it makes it less permeable. The membrane becomes less permeable. It's bigger. It pushes the molecules aside, gives more space, so it keeps the fluidity of the membrane while stabilizing it in general, because it also sticks to the phospholipids that are near it. It kind of pulls them together, but it's still big enough that it increases the space between the molecules as well. So it's kind of a crazy molecule. You guys are having a nice conversation. I hope you listened and wrote down what I said, because it will be on the test on Wednesday. All right, does that answer the questions then? Yes, okay. Let's do it now. Embedded proteins are often found spanning the membrane of a cell or organelle. These proteins serve as channels for specific molecules. So this is an extension question, because this tries to answer this question. If you have a cell, remembering that the cell is made of what? The membrane is made of what? It's made of phospholipids, just phospholipids, or how many layers? It's a bilayer. It's 2 layers, right? It's a bilayer. You have a problem, don't you? If your nucleus is here, whether you're prokaryote or eukaryote, you have a problem. Should we keep it a prokaryote? Should we make it simple and keep it a prokaryote? Let's do that. So a prokaryote only has one membrane. It has to, so it exists, and it multiplies. It has a single chromosome, a double helix, single chromosome, sitting in the middle somewhere. It's little plasmids. Has ribosomes in the cell. Right, ribosomes. Has some structure. There it is. What's the problem with having a bilayer just like that? Just a bilayer.
Shouldn't have cell wall.
Oh yeah, it does have a cell wall. Very good point. Bacteria do have cell walls. They have different cell walls from plants, but they do have them. Someone want to close the door, please? I'm not dealing with that. They make the cell wall. The wall is on the outside of the cell membrane. They make it. Okay, so you got a cell wall. You got a membrane. Cool. What's the problem? You remember it's a sphere, right? It's not a flat disc, so it's a sphere. What do living things do? Let's go with that. All right, they reproduce, correct? Hold on a second. Let me move along. They reproduce. They react to their environment. I guess this would be one of those very important main ideas, right? They reproduce. They react to their environment. What else do living things do? Grow. They grow to some extent. They grow, I guess. You start growing. Growing becomes a problem for single cells. They do get bigger, but there's a very quick limit. They don't get. It's not like you're going to get a giant cell, right? So there's a limit to size of cell. Yeah.
Oh, they need energy.
They need energy. Right? So they consume. They need energy. They consume energy, right? If they're going to move, if they're going to do stuff, even cells like this, bacterial cells like this, they don't actually swim. They are moving things in and out. They're moving ribosomes around. They're moving things in here, so they need energy. Probably a lot less energy we do, but still. What else do living things do? Yes, go ahead. Anybody?
Maintain
They evolve adaptations.
Right, they do evolve. Very good. And somebody said, what did you say a minute ago?
maintain homeostasis.
Yeah, maintain homeostasis. What else? Yeah. Make waste. They produce waste. They eat. Where did they say? Did we say eat? Need energy. So let's say in parentheses eat and waste. Whatever that looks like. Can we say breathe or exchange gases? It's a pretty good list, isn't it? So those are kind of main ideas of what life is. If we want to know the difference between that table and life, it's a lot better than the ancient Greeks did as far as understanding what life is. Right? Because, they looked around, they said things that live move, right? They move. Even rocks move. So if rocks move, they're alive. Okay, that's fine. But we have a much better list here. What's the problem, though, with what I've drawn right here, with this model? What's the problem with this model that we have here of this fluid dynamic cell membrane producing a cell wall in a round form, floating in some spit or in their intestines or sitting on the table? What's the problem with this cell? Look at these characteristics and tell me what's the issue. I'll wait. And why don't you discuss it amongst each other and come up with an answer for me? I'll wait. I'll just sit here and drink my coffee. You guys discuss. There's a problem here with this model.
So this is bacteria cell? Which one doesn't? You said this is a bacteria cell or animal cell?
It's a bacterial cell, but it doesn't matter what kind of cell. No matter what kind of cell, to have a membrane and a cell and or a cell wall, whether it has a cell wall or not, right? If it has a cell wall, it's not an animal cell. Make sure you know that, because you will see that repeatedly. If you have this cell, it has a membrane that separates the outside from the inside, allowing for better organization inside. But what's the problem when you look at those characteristics and just what we have drawn so far? Sophia? Say that again? What about the DNA? Oh, it's there. The DNA's there. I didn't make it clear. I'm sorry. The DNA in a bacteria is round, and it's a double helix, but it's round. So that's actually a DNA. And so is this. DNA in bacteria is round, and prokaryotes are round. Yeah.
Compartmentalization is like not.
Right, but even if it was compartmentalized, even if this was a eukaryotic cell, prokaryotes are not compartmentalized, but they live. So we know they're alive. So we know they do all those things. But if we look at this cell that I've drawn, this model of just this cell membrane with phospholipids, maybe some cholesterol in it, and a cell wall, there's a problem with this list of factors. You can't do 2 or 3 of these. Based on what I drew on that board, you can't do 2 or 3 of these. Which one is going to be the biggest problem for this cell that I've drawn on this board? Yeah.
Maybe needing energy.
Why?
Because it needs chloroplasts
No. Good try. No, it does not. Plants. Chloroplasts are found in plants. Chloroplasts do make energy. You're right. For plants, they do collect the sun's energy and turn it into glucose, help turn it into glucose. So that's absolutely right. But there are other ways of getting energy. For instance, you don't have chloroplasts, but you get energy. You eat it.
Yeah.
But let's say, no matter what, what's the problem? There's a problem with 2 or 3 of those bullet points. It is getting energy, so it is getting energy is a problem. So, but what is that problem? Yeah.
The gas, the exchange of gas.
Exchange of gas is another one of the problems that this drawing is going to we're going to have issues with, or at least theoretically we might have issues with. So, needing energy, and it's going to be hard for what we've drawn so far. Anybody guess why? Yeah. That's right. It's how are you going to get things across? The whole point of a membrane is to separate inside from outside. So as I draw these, whether these are phospholipids, bilayer with or without cholesterol, you need to get stuff inside and outside the cell. How are you going to get stuff in and out of the cell? What is this? This is a membrane. What's the membrane do? That keep things in and
things out.
it keeps things in. In and out. It separates. If the job is to separate, then that's going to be a problem for needing energy, and it's going to be a problem for exchanging gases. Do you agree? Yeah. How do you exchange? How do you do it? It's the whole point is you're separating. So this is not an impermeable membrane. This is not plastic wrap. Membranes are not plastic wrap. They're not waterproof. That's not what they're made for. You are not waterproof. Yeah. Yeah, that's a good point. You could also say it would be hard for it to maintain homeostasis if it can't let things in and out. Thank you. Set it down. So how do you get things in and out of the membrane? That's the question. If you have this cell, how do you get things in and out? Well, the key is embedded proteins. Proteins that you embed in the membrane. So if you just have a membrane, and I'm just going to go ahead and draw just the membrane this way if you don't mind. I'm not going to draw the phospholipids. Oh, what the heck, I'll draw them. If I have just that, then that's an issue. Because anything, you all know you need salt. Is that correct? Yeah. So you have sodium ion and chloride ion and calcium ion. They have to get in. These are ions. They will not get past the lipid bilayer. So you got a problem. Unless, and we're not going to talk about what kind of transporter it would be. There's different kinds of transporters. We'll talk about that soon, okay? But right now, let's just talk about this membrane. You need to be able to get these ions across this lipid layer. The lipid layer won't let it through. They're nonpolar. These are ionic, very difficult for ionic compounds to get through the lipid layer. Small polar molecules like water could get through, maybe. They do get through slowly. But even water, you need to be able to get water through the membrane faster. You need to get through the membrane faster than you would normally without this help. Sometimes you need help to get across the membrane. We don't put holes in the membrane. Why don't we just put holes in the membrane? Just let anything in now. That's right. Anything getting out. And then the problem with that is you can't maintain homeostasis. How do you keep organized if all you do is just have a hole that lets anything in and out? Well, you don't. So that doesn't work. So holes don't really work that well. So what you do is you make specific channels. Look, it's like an open window. You want to open your window because you want air in the house, right? But you don't want the flies to come in, or the bees, our famous bee that keeps flying around here. So what do you do? What do you do? Yeah, put a screen up. Open the window, but keep a screen. You need a window. You need to be able to get in and out of the house. You need air in, you need light to come in, you need to be able to go out. So if you don't want the cold air to come in, you close the window, but you have light. You let light in by making the window out of glass, right? So you have allow for some things to get across that separator that we call a wall, right? Or we allow things in and out through windows and doors. So the membrane needs windows and doors, and we call those windows and doors proteins. And we make them with proteins. Now this is more accurately drawn because it's just a chain of amino acids, all right? A chain of amino acids that wrap around and form coils. So you have, and part of the amino acid chain, remember it's a chain, so you have amino acid 1 next to amino acid 2 connected through peptide bonds. So if you don't remember that hurts, right? So I wish you would have, I would say at that point, you have to go back and look at what it is what exactly is an amino acid and what is a protein. I can't go back and do it now. You can go back in your notes, go back in the videos, or you can see me doing tutoring. But if we look here, these amino acids have what kind of R groups? What kind of R groups do you think these amino acids have? The ones that I'm drawing right now. In yellow. What kind of R groups do you think they have? Somebody make a guess. Nonpolar. Perfect. That's exactly right. And these that stick out, what kind of R groups do you think they have? Polar. So the polar R groups, those amino acids stick out. And the nonpolar, those R groups get stuck in the membrane. If you have a hard time remembering what amino acids, again, they look like. And so but that's an issue you're going to have to fix. So what they do is they stick in the membrane like this, where the nonpolar parts are here in the middle, but they don't necessarily, they're not just solid proteins, right? They can create little channels. We call them channels. So if we took a look at that, and from the side, it would look something like, if we just cut it open, then there would be a channel here that would be polar, an opening here, and then the sides would be the amino acids that are nonpolar. So this is cut, and then out here would be the amino acid chains, R groups that are polar. And that would allow is that now you have a channel that allows calcium to flow through it. Yeah.
It's 9:14.
You can go. Yeah, you can go. I'm recording this so you can see it when I post it. So that would be one type, and we call this a channel protein. There are other kinds we will talk about. So this is called a channel protein. Allows things in and out. All right. Which is what this is doing. This is a better drawing because this is more like it's a chain of amino acids. It's just all the way around. It's creating a tube. It's creating a tube. I'm drawing a cut piece of the membrane, but you have to imagine if you were looking at a bunch of amino acids from the outside, a bunch of phospholipids, a ball of phospholipids, which you would see from the outside, if you're looking at the cell from the outside, all the phospholipids would be moving and oh. There you go. There. But there would be these proteins that are embedded in the membranes that are hydrophilic or polar around here, around this part that sticks out, but they're hydrophobic along the center, along the inside. They're hydrophobic on the inside, but it's a tube. It's literally it is in a way a hole, but it's just a channel that only allows that ion through because of its shape. Its shape and constituency only allows ions through. And then what can you do with this protein? You can put them in or take them out. You're constantly putting these proteins in, constantly taking out of the membrane. You need more calcium in, you put the channels in. You need less calcium in, you take them out. And we have other control mechanisms as well. There's some that are voltage gated, some that are ligand gated, and we'll talk about those later. All right, but the bottom line is that's what you need. You need proteins. So let's go on and read then what this says. These proteins are channels specific to notice what it says. These are channels that are what? Specific for that molecule. So they're specific for that molecule. They travel through the membrane, either into or out of the cell. What sections we already answered these? Yeah. Yes, absolutely. We already answered this one. What sections are embedded in the protein chain are most likely to contain amino acids with hydrophobic R groups? The section that what?
That's inside?
that's inside the membrane, in the lipid part of the membrane. Why? Because like dissolves like. The hydrophobic R groups would mix with the lipids. What section of the embedded protein is more likely amino acids with hydrophilic R groups? The parts that's sticking out, the membrane. That makes sense. Inside and outside the cell. What sections of the embedded chain are most likely to contain amino acids with hydrophilic R groups? Oh, I already answered that. Sorry. Some membranes have surface proteins on them. So notice this is a surface protein. You've heard of ABO blood groups?
Yes.
Everybody's heard of A. Who's A blood? Who's A type blood in here? Just A. Okay. Some of you may not know. Who's B type blood? All right. I'm O. O positive. You're O as well. O. Right on, sister. A's and B's suck. O's are best. So just so you know. So O is the most common, interestingly, but we are universal donors. O means we don't have surface proteins. These don't exist for us. Okay. Not these surface proteins, the blood type. We have other surface proteins, but the blood types are identifiers. They identify you. We can give our blood to anybody, you and I. O negatives especially. I'm O positive, so I can give my blood to almost everybody. We're givers. AB blood groups are acceptors. They can accept blood from anybody, but they can't give it to anybody other than AB. We can talk about that later. But I just want to mention it. All that, ABO, AB, AO, BO, no, not body odor. OO, all those blood groups are just cell types, proteins outside, hanging on the outside of the cell that identify that cell as being part of you. If you have somebody with A blood gives you blood and you are B blood, this surface protein is going to have a different shape. Your body's going to say, that ain't me. It has to die.
So if somebody, if you get the wrong type of blood, can that hurt you?
Oh yeah, it'll kill you. It turns out some people could take it one time, but after that you become sensitive to it. It's like having it's having an allergic reaction. That's what it is. It's an allergic reaction. And you're having allergic reactions are your body's way of killing anything that doesn't belong to you. And you can be allergic to yourself. It's called an autoimmune disease. It's not like things like MS, things like lupus are diseases where your body's attacking itself because it's confused. And we'll talk later, and this is much later, we can talk about why that is. But when you're talking about ABO blood groups, you're getting blood from another human. If that blood doesn't match your blood, your body's going to say, this doesn't belong to me, I'm being attacked. It's a cell that doesn't belong to me. What is the body going to do? Kill it. It's going to have an allergic reaction. And it's not an allergic reaction, so a peanut touched my skin, so I get welts. No. This is an allergic reaction where it's in your blood. It's happening all over your body all at once. You're dead. You have a major allergic reaction throughout your whole body and you die. So you cannot take blood that doesn't belong to you, or at least from your group. And no, it's not race-based. So right away I can see people thinking, oh, my group. No. ABO blood groups have nothing to have very little to do with race. Yeah.
Very little.
I say very little because there are some slightly different percentages of different blood types. That's to be expected because it's an inherited trait in different populations. But across all races, there is no difference. But in populations, like in family groups, you'll get more predominantly this type of blood or that type of blood type. Blood type. Yeah.
So if you were to take your blood and like when somebody was sleeping, you gave it to them and they died, the people who
The coroner would know that happened, yes. It's obvious what happens. Hey, there's a lot of crime shows out there. I'm done watching it. The British are really evil they make really great crime movie shows. And PBS has a fantastic bunch of good crime shows. PBS, a lot of great crime shows. Yeah.
Okay, so let's say I'm like AB something.
Uh-huh.
And you're O positive. I can give you blood. But
No, opposite. I can give it to you, but you can't give it to me.
Okay.
So O blood type can give to anybody. We're universal donors. AB are blood universal acceptors. You can accept because you have A. O means we don't have any surface protein identifier. That's what it means. So our blood doesn't have that surface protein that says, "Yo, I'm not you." But A has a surface identifier that's A. So I have none. The way my body knows, it checks, by the way, that we have cells that go around and literally, they don't have eyeballs. They touch and they see, oh, this protein's not good. Okay, this is mine. So if there's nothing there, if it's smooth, we're ready to go. But as soon as it sees an A, it's like, oh, karate chop to the face, it's dead. So if it feels a B, also my body's gonna say no, because I shouldn't have any. I shouldn't have A or B. But if I have, and if you're AB and you give it to me, then my body's gonna go, no, you're not O. Now if you're AB, you have both. If I give you, I don't have any. If I give you blood, is there anything there for your body to react to? There's nothing there. There's no bump. So it's like it doesn't exist. If I give you blood and you're B, same thing. If you're AB, then somebody with A can give you blood because you have A on your cell surface. It can't tell the difference if it's A or AB. There's two proteins on your cell surface. There's an A and there's a B. So she's A, she gives you blood. Your immune cells read A. That's cool. I got A on my cells. He gives you blood, it's B. Your cell reads it. Oh, I got B. That's cool. I give it to you. It's O. No, there's nothing there. I'm cool. So AB can accept blood from anybody.
But I can't give it to you.
You can't give it to anybody except people that are A and B.
Okay.
Yeah.
What's the difference between like positive and negative, like O positive?
Good question. It's just a tiny surface protein, smaller surface protein. It's still a surface protein. It's smaller and not as and they're not the three main groups. So there's two types, and there's not three different versions of the gene. It's just one. Either you have that gene or you don't have that gene.
And like, what's the difference between A and B? Like I don't know, because I know that they're like.
So they're different. Just think of it this way, and we'll get into more of this when we get into genetics, okay? But for now, just think of it this way: it's two different shapes. The proteins have two different shapes because there's two different genes, alleles. They're the same genes. But bless you. Cover nose to elbow to nose for Pete's sakes. If you got any snot on my iPad, I will immune cell you. Anyways. What was I saying? So genes are inherited. Genes produce proteins. If you there's two versions, two alleles. Actually, with ABO blood groups, there's three alleles. Three versions. There's a version that's an O, there's a version that's A, and there's a version that's B. So you have three alleles, same gene, same locus on the same chromosome. So you inherit either an A or an B. They produce three different proteins. One protein, there's no cell surface. Maybe it's all hydrophobic, maybe it's not produced at all. I don't know, but there's nothing on the cell surface. So there's nothing to read. Okay, that's O. A blood type, maybe it's a triangle. I don't know. It's a weird shape. It's not a simple shape. It's complicated. But let's call it a triangle. Okay? B is a circle. All right? So two different shapes.
Yeah.
So you inherit two genes. One from mom, one copy from mom, one copy from dad. If dad gave you an A gene and mom gave you a B gene, you're AB. So you have triangles and circles on the surface of your cell. If mom gave you B and dad gave you B, you're BB. You're B. All you got is circles. Mom and dad give you A, you're triangles. And if mom and dad give you O, you have nothing on the cell surface. That's it. And which one you have, you react to the one is not you.
So like, if you had like an A and an O, like would it be?
The A,
Oh,
because O is, there's nothing on it.
oh, yeah.
But this-
okay.
could you still
So
get
O
blood from
equals
an
like
O person?
0?
So that's the trick. A lot of people leave. We'll talk about genetics. But I've heard of stories where kids are in class and they learn about inheritance, and they think, well, O is recessive, right? It is. And so the only way you can be OO is if both parents give you O. But mom's A and dad's
So
O.
it'll be A.
Oh my God, mom's not my mom. Because I can't have, if mom's A and I'm O and dad's O, I have to be A blood type if I was mom's baby. No, because mom can be AO. So mom could have given you an O, even though she's A. Because there's two versions, one is O. So the details matter here. But that's in genetics, we'll talk about that later, okay? Yeah.
You could have separate like blood type letter from nowhere, like even if one of your parents.
Could you mutate? Could you theoretically you could your cells you could have a mutation that's a new blood type, or that is a blood type that the mutation could happen that would make you mutate you to a B. It's highly unlikely. So if your blood types don't match, more studies would more study would have to be done. But it's likely that it's one of the two people are not your parent. That is true. That's how they used to do paternity tests, ABO blood groups. It's not very reliable because of all these different options, by the way. Yeah.
So, does blood type anything to do with like gender?
No gender. It's just a blood. It's just there's no gender, no issues. It's just a cell surface protein. It's literally just for IDing you. It's your ID. It's your license. It's your driver's license to be in your body. If you don't have that, your body will kill it. That's it. Yeah.
When will we be starting our genetic fusion?
Huh?
When are we going to be starting our
As soon as we can get through this stuff. So let's get on with it. So this says some membrane proteins are surface proteins, as we say, ABL blood groups. These proteins often serve as signaling function between cells. Purpose and mechanism by which the surface proteins are able to attach to the membrane. So how could they attach? What must they have here in order to be stuck in the membrane? They have to have a what? They have to have a little tip that's what? That's nonpolar. If it's going to be embedded in the membrane, it has to have some nonpolar piece. Otherwise, it won't stick. It'll just float away. And obviously, it needs to have a polar piece as well that would stick. This would all have to be polar. But then, if it's going to lay down like this, it probably has maybe a little bit of a nonpolar tail here and here. Otherwise, it would just stick out and float around in the water because water will dissolve it. So if it's going to be folded this way, it's likely, and you might even have some nonpolar pieces on the inside here. Just for the record, you might actually have maybe some nonpolar insides here as well to make it fold this way. It's a folding. It's all about folding, like making a paper airplane or making a little paper goose or paper flower, right? It's all about taking that protein and folding it in a certain way to get a specific shape. That's what we're talking about. And the shape leads directly to a function. Are we all good with this? You look like you're in pain. Are you okay? Okay. All right. Well, let's go on and you guys do research. Let's answer this last question. What does cholesterol do? Go ahead and figure it out. In small groups or on your own, and I'll expect an answer in 5 minutes? We want to hear first from our most majestic and interestingly quickest group to find the answer. Go. Say your full name and then state your answer for the record.
My name is Mayar Abdulhadi, and I think that the cholesterol's main function for the cell membrane is to stabilize it because it's a nonpolar molecule and it stabilizes the cell membrane. And it's a lipid.
So close.
yet so far.
It might not be the answer you're looking for, but
Yeah,
it's not
but-
incorrect.
Oh, it is incorrect, at least in part. It's in part incorrect, yeah.
You just wanna get.
So close and so far. Oh, you asked for it. Alright, you totally answered.
No,
You
you
sat there and
offered.
talked about politics.
Offered.
You talked about politics for 5 minutes. So the cholesterol has a polar section. It does have a non-polar, it does have a polar part. Let's take a look one more time at cholesterol really quickly. Oops.